Yeah, hello everyone. I extend a warm welcome to you and express my heartfelt gratitude to Big for giving me this opportunity to address you today. First of all, I will introduce myself. My name is Dinesh Suminda from Sri Lanka, and I'm currently pursuing my PhD at Jeju National University. It is indeed a privilege to share my research with you today. We are exploring deeply into the critical issue of environment pollution and its impact on public health, particularly emphasizing the investigation of arsenic in the development of osteoarthritis. In this research, we will uncover the hidden danger of arsenic, and I name that as a silent threat in our environment. Let's explore this together. And I will go through in this content introduction, material method, results, and conclusion. First, I will start my presentation by providing an overview of the current status of environment pollution. We will go through the we will go through the latest statistic and report that reveal the alarming extent of pollution caused by the human activities, from industrial growth to urban expansion. These activities have significantly increased the pollution, causing harm to our ecosystem and wildlife and also human health. For example, according to United Nations, CO2 emissions have surged nearly 50% since 1990. And also WHO report that 90% of us breathe polluted air. And most importantly, the contaminated water cause millions of deaths annually. You can imagine from the bar chart that I present you from the recent published paper how crucial this situation is, because total pollution kills people more than any other factors. And when the global population set to exceed 8.5 billion by 2030, the challenges will rise. When we see into this pollutant, one of the common factors is heavy metal like lead, cadmium, mercury, and arsenic, which persist and also accumulate in our environment. We all know their sources, including industrial waste and mining activities, and how they infiltrate our water and also soil and air, and understanding how we encounter this pollutant through food, air, and water is key to grasping their full impact on our health. Next, we dive deep into the world of heavy metals and their significant effect on our health. It is fascinating that a large chunk of environment research, about 1/6, is focused on these metals and highlighting their danger as an environment hazard. Let's see how this metal can disrupt our bodies and acting as a neurotoxin and carcinogens and disrupting the hormonal system and also circulatory system, and their tendency to accumulate over the times make them silent yet persistent. It is leading to long-term health issues. And also in this segment, I particularly focus one of the unique nature of articular cartilage, the smooth tissue at the end of the bone. This tissue is special because it does not have its own blood supply or lymphatic drainage, and it's made of 60 to 80% water. And it is maintained by the homeostasis of internal factors and also external factors like environment toxic. It can irreversibly damage the cartilage. This is crucial for understanding why heavy metals, especially through environment exposure, can affect the osteoarthritis. We will then move to discuss how so far the metal influence on OA, looking at metal like zinc, and mercury, and also cadmium and selenium. You can see in the slide the cadmium and also selenium and also hexavalent chromium previously studied and explained their effect on cartilage health. However, our polluted environment making multiple challenges, and these are on the getting more highlighted in a way. And these challenges make our research extremely relevant and also urgent. Focusing on arsenic, we will explore the link with osteoarthritis, especially in those with the occupational exposure. In here, we present the data from 1990 to 2019, showing an increase in osteoarthritis cases parallel to arsenic exposure. The correlation is very similar. In this spotlight, arsenic as an invisible aggressor. And this data actually is sourced from the Global Health Data Exchange database. We also discussed the widespread arsenic contamination in groundwater. You can see in the world map how arsenic distributed, and especially in Asia, over 90% of population affected. And if we consider, especially in Korea, 42% of groundwater have arsenic level above the World Health threshold level. I mean, above that one. This highlights the global scale of arsenic contamination and its potential health impact. So let's move to what is osteoarthritis. I mean, it is the most common form of arthritis, and it's caused by the imbalance of anabolic and catabolic factors. You can see in the figure, when anabolic and catabolic factor balance in seesaw, maintain the healthy cartilage. But when it is unbalanced in the cartilage, degeneration and OA symptoms appear. In this slide, we can see the arthritis prevalence and also anticipated increase. 53.2 million US adults have some form of arthritis. And from that one, 32.5 million has the osteoarthritis. And also they are spending around 5,000 million US dollars annually for osteoarthritis treatment. Finally, I will propose the hypothesis that arsenic exposure can disrupt the anabolic and catabolic imbalance in our joint, leading to osteoarthritis. For that, I have designed my experiment according to this one. So let's move to material and method. Actually, we start our journey with in vitro studies, focusing on primary cell culture techniques. In here, we closely examine the cellular response to various conditions and treatments. Those are influenced. We check by the using OCR and FACS and RT-PCR and QRT-PCR techniques. Especially in here, I want to emphasize we employ chemical inhibition and also siRNA gene knockdown method to discover the pathway involved in catabolic and anabolic factors in the cartilage cells. Shifting to our in vivo studies, we follow a systematic approach involving surgical operation on animal models. It includes the eye injection, I mean the intra-articular injection, and also sham surgery and the DMM surgery. These procedures stimulate the conditions of osteoarthritis under the arsenic exposure. In our methodology, also surrounded by the administrating arsenic through drinking water experiment. This setup aimed to mimic the real-world exposure scenario, and histopathology study were conducted at various time points, including 3 weeks and 8 weeks experiment, and also accumulation of the metal ion quantified by ICP-MS method. Basically, DMM surgery full form is destabilizing the medial meniscus. You can see in the meniscus in between two cartilage, I mean the femurs and tibia. In that two bones in between the cartilage, we can find the medial meniscus, and it absorbs the pressure and between the two bones due to weight. And during this surgical procedure, we operate the medial meniscus and start the aging process in young mice. You will more understand it further when we are presenting our data. Let's move to result sections of our presentation. In this research, one of the key findings is in fact on arsenic on mitochondria function in chondrocytes. Actually, in mitochondria is the vital powerhouse of our cells, crucial for energy productions and also stress resistance. This provides the critical insight into how arsenic jeopardizes the cellular energy dynamics within the joint cartilage. We initiate our experiment with detailed examinations of mitochondrial dysfunctions using the seahorse mitohysteresis assay. This explains the fascinating aspect of energy demand and proton leakage and also mitochondria ATP productions, particularly in the chondrocyte treated with arsenic. This phenomena indicate a compromised mitochondrial membrane, leading to insufficient energy productions. The increased energy demand coupled with the reduced efficacy of mitochondria, creating a challenging environment for maintaining the homeostasis. Based on these foundations, we further examine the impact of mitochondrial changes. We employ JC1 assay and confirming the significant mitochondrial potential impairment in arsenic-treated chondrocytes. This vital finding link low membrane potential increase the ROS productions. However, treated with ROS scavenger NAC remarkably recover the mitochondrial membrane potential damage caused by arsenic. And also our experiment did not stop here. We measure the intercellular ROS level in chondrocyte with and without the NAC using fluorescence image and also flow cytometry quantifying. This highlight the arsenic alone elevate the cellular ROS and NAC significantly mitigate the this effect. Next, we figure out the bridge, the gap between mitochondrial dysfunction and also metabolic alteration in chondrocytes. Employing RT-PCR and qRT-PCR analyze, we reveal a dose-dependent decrease in anabolic genes like COL2A, SOX9, aggregan, and increase in the catabolic gene like EPAS1, CYP8, MTF1, and also MTF2 genes, and also, metalloproteinase gene, MMP2, 3, 9, 10, and 12 and 13 genes, or expression under the arsenic stimulation. These results reveal that arsenic can disrupt homeostasis by regulating the anabolic and catabolic balance. After that, we took an exciting turn when we examined the aquaporin channel family role in the metabolic seesaw. We decided to study about the aquaporin channel family because arsenic can penetrate the cells through different aquaporin channel in different tissues, especially a PQ9 ion channel highly expressed in the chondrocytes and responsible for arsenic influx. So we found that disrupting APQ9 importer with the fluorectin chemical inhibitor not only restore the negatively regulated anabolic genes, but also temper the catabolic gene expressions. From this data, we can strongly confirm that arsenic is the only factor that is responsible for this unbalance. Our in vitro finding paint a clear picture of how arsenic disrupts the chondrocyte metabolic balance. Then we try to confirm it with the mice experiment as well. So we inject the 10 microliter of 100 ppm concentration of arsenic oxide directly into the mice knee joint for 3 weeks by the once a week of eye injection. We saw visible osteoarthritis development by injecting arsenic into a knee joint. You can see it on the safranino staining images. And in this image, you can see the cartilage in the red color, and also arsenic injected knee, the red color cartilage clearly degenerated. And also we can see the synovial inflammation in mice after arsenic injection. And all of these safranin staining slide were score according to the OCR grades. And another important point of our study is based on our in vitro data and the prior identification, the HIF2 alpha and CYP8 acting as a main cellular catabolic mediator. And overexpression of HIF2 alpha in the chondrocyte causes significant increase in CYP8 promoter as well. So HIF2 alpha is a transcriptional activator that play a role in regulating the mitochondria oxidative stress response and also activate numerous downstream genes. Therefore, we have confirmed CYP8 and the EPAS1 overexpression in our 3 weeks experiment. You can see it in the immunohistochemistry slide. CYP8 and also EPAS1 highly expressed. And subsequently, in another group of mice, after 3 weeks of injection, we kept another 5 weeks to observe the various OA symptoms such as cartilage degeneration and also osteophyte development and the sclerosis and high synovial inflammation. As we predict, we could see the severe symptoms after 8 weeks of experiment. You can see in the slide large osteophyte development and the sclerosis. And proteoglycan barely express at the end of cartilage deep zone area. So we did not stop in these mere observations. We try to understand the effect of arsenic on cartilage degeneration, especially through contaminated water consumption. We administrated 10 ppm arsenic contaminated water mixed with the normal drinking water for 16-week duration. After 8 weeks of arsenic consumption, DMM surgery were conducted, and after 8 weeks later, histological analyses were performed. And our findings clearly indicate that DMM surgery mice in arsenic contaminated water group are showing the shocking cartilage degeneration and also synovial inflammation compared to normal water supply mice. We further confirm that HIF2 alpha and CYP8 also responsible for drinking water cartilage degeneration by the ISC data. And one of our most striking finding was the significantly accumulate arsenic in articular cartilage of the mice knee area. For the first time, we found that arsenic can accumulate in the cartilage tissue through drinking water. We quantify this accumulation by the ICP-MS, and interestingly, despite of this accumulation, other organs remain largely unaffected. This statement proves the mice body weight unchanged and also HNE staining tissue examination. Shifting for our molecular mechanism, we examine the effect of two main important downstream transcriptional factors, HIF2 alpha and CYP8 level on cartilage homeostasis. Here, siRNA knockdown experiment play a crucial role, revealing the potential for reversing the arsenic effect on anabolic and catabolic genes in mice chondrocytes. You can see in the slide. And also, we move to the upstream factors. We try to interconnect ROS generation and also HIF and CPAID signaling cascade. ROS generation was abolished by the NUC scavenger, as we did in the mitochondria assay. But in here, we check how it affect on gene regulation of anabolic and catabolic factors. From these data, we can purely predict that ROS triggering from upstream to start signaling to the osteoarthritis progression. However, ROS, CYP and HIF2 alpha interconnection is not clear. Therefore, we try to dig into the pathway analyze and find the exact interconnection. Then we find out that stress surviving factor, AKT, mitochondrial stress, and also downstream factors like HIF2 alpha and CPAID and cartilage degeneration interconnected in OA studies. Therefore, we investigate whether AKT signaling are involved in arsenic-induced cartilage degeneration and metabolic imbalance. By inhibiting this pathway, we observe noticeable reduction in catabolic and anabolic gene expression and also protein level expression as well. From this data, we can confirm the crucial role of AKT in arsenic-induced cartilage degeneration. However, AKT cannot independently activate the HIF and CPAID transcription factors. For these reasons, depend on prior investigation, AKT in control of NF-kappa B, and it's responsible for activating the CPAID promoter region and HIF2 alpha transcription factors. Therefore, we investigate whether NF-kappa B signaling are involved in arsenic-induced cartilage degeneration and the metabolic imbalance. By inhibiting those pathways, we observe noticeable reduction and increase in the catabolic and anabolic gene expression and protein level subsequently. At the same time, we chemically inhibit the HIF2 alpha to examine the catabolic and also anabolic factors. And here is the key finding in our research. The arsenic disrupt the homeostasis in chondrocytes, and also consumption of arsenic contaminated water lead to arsenic accumulation in the knee joint, and arsenic accelerate the both early onset of OA symptoms and the post injury OA, and arsenic induce ROS generation and disrupt the mitochondrial functions, and HIF2 alpha and CPAID were involved in this process. So those are the key factors we can take from messages. And as a conclusion for our presentations, our finding are consist with the statement that occupational arsenic exposure has increased OA prevalence all over the world. And as a result, our discovery revealed for the first time that arsenic disrupt cartilage homeostasis, eventually leading to OA development through the ROS dependent HIF2 alpha and CPAID axis via the AKT and NF-kappa B signaling. So I would like to extend my heartfelt thanks to Professor Yannockson for his support and expertise for bringing this work ahead. And also I would like to express my sincere gratitude to my team members. And thank you everyone, and thank you for your attention. I look forward to hear from you.